Hi, my name is Dominic Carrera, and I am a wildlife ecology and conservation major here at UD with the Unique Strengths program. This summer, I have been researching the prevalence and diversity of avian malaria parasites in birds throughout the United States. Most of my research has been conducted in the lab of my faculty mentor, Dr. Vincenzo Ellis, whose work mainly focuses on the evolution and ecology of diseases in all sorts of wildlife. For the purposes of my research, I focused on one group of birds in particular, known as raptors or birds of prey. This group includes birds like vultures, owls, eagles, hawks, and more. As for the avian malaria parasites I am studying, they belong to an order of parasitic protozoans, known as hemosporidians, that look like this. This is an example of a Hemoproteus hemosporidian infecting a bird red blood cell. The two other kinds of these parasites that I look for are known as plasmodiums and leukocytosins. As you can probably guess, avian malaria can be just as harmful to birds as their own kind of malaria is to us. And with temperatures rising from climate change, avian malaria is one of the many diseases we may unfortunately see expanding in range. This is because higher temperatures can allow disease vectors, like mosquitoes, to reach new places and infect new hosts that often have less immunity. This poses a serious threat to many of our bird populations, as avian malaria impacts all kinds of birds, from the raptors to robins to chickens to even penguins. So in order to better understand this disease, the bulk of my research involves the core component of all life, DNA. In particular, it involves screening for the DNA of avian malaria parasites within these kinds of raptors. As such, the first step we take is to gauge how much DNA is present in each sample. This is done by taking a small amount of the sample and pipetting it onto the nanodrop machine. This machine then shoots a beam of light through the sample and records the amount of light absorbed by the DNA to determine the concentration. After the DNA concentration has been determined, we then use two different PCR or polymerase chain reaction techniques to screen and confirm the presence of the parasite's DNA within each sample. Once the PCR techniques reveal which samples are infected, the samples are then sent to be sequenced. Sequencing is done in order to know the exact order, or sequence, of the nucleotide base pairs in the parasite's DNA. Knowing this sequence allows for the identification of the parasite species present. Information like this is foundational to better understanding the ecological impact of these parasites, and for knowing which are the most problematic. So far, we have screened 91 samples, with 36 testing positive for at least one of the three avian malaria parasites. The disease also managed to infect roughly half the species present in the dataset, with hawks and owls being among the most infected. I'm excited to learn and share more about my results once the parasite species present have been sequenced. I've been so grateful for this opportunity and hope my research can contribute to a better understanding of this disease and help birds all around the world.